Hello, my dear children. Namaste and welcome to session 3 of our class 10 ICSE biology chapter genetics. Yes, and finally, after two rounds of, or rather, two sessions of giving you the basic concepts of this chapter, today we are going to dive right in to the hardcore concepts of this chapter, which many of you think is very hard. But trust me, it's not. Stay with me for the next 30 to 40 minutes and let us learn this together and let's see how easy this is after all. Okay? But as always, before we get started, let me tell you something positive so that you can carry this forward for the rest of your day, for the rest of your week, whatever. You only fail when you stop trying. So just because one particular incident happened because of whatever reason, that doesn't mark you as a failure in life. The more you try, the more your chances are for success, right? You only fail when you end up stop trying. Let's all remember this and move forward positively. Okay, so today we are going to be learning about monohybrid cross, dihybrid cross, and the three main Mendelian laws, and of course their significance also. So yes, Mendel, remember I told you in the last session, Mendel was an Austrian monk and he was very passionate about his pea plants. He loved his pea plants like his own children, in fact, but he was a very serious scientist. So he counted a lot of pea seeds. And also, I think I told you this also, that he was also an expert mathematician. So he applied math and statistics and all of his basic senses of logic to biology, or rather whatever he observed in his pea garden. Okay, so let's look at what all magic came out of that pea garden. And this, let me just recall that also for you. This happened around the mid 1800s, around 1855 to 1865, or around that period. Okay, so let's get started with monohybrid cross, which I have defined for you in the previous session. Breeding between two plants differing in traits of a single character. So Mendel started off his experiments in the pea plants through what we call the monohybrid cross. So basically what he did was, remember I told you he chose 7 different characters, right? 7 different characters which contain 7 contrasting pairs of traits, right? So he went about examining one pair of trait at a time. So to study one pair of traits, or rather one character in the pea plant, what he did was he picked up 2 pea plants which were similar in 6 of those 7 characters but differed only in one character. Okay, he did this, he repeated the same set of experiments for all the 7 characters or all the 7 pairs of traits. So for our understanding, let's pick up 1 or 2 random pairs of traits among Mendel's characters. But the results were all the same as I have told you in the previous session, so it doesn't make much of a difference. All that matters is you understand how exactly Mendel came to his conclusions. Okay, so yes, first of all, before we get started, in a monohybrid cross, remember I told you the reason Mendel chose the pea plant, one of the reasons was that it's got bisexual flowers, which means it's normally self-pollinating, but it can also easily be cross-pollinated. Which is important for a scientist because he wanted to try out different combinations of plants, like not just a cross between the anthers of a tall plant, the pollen grains of a tall plant with the egg cells of the same tall plant, or the pollen grains of a dwarf plant with the egg cells of the same dwarf plant. No, he wanted to try and crossbreed them. He wanted to try and cross-fertilize them. So how exactly is artificial cross-pollination done? Here it is given to you step by step. First of all, of course, he chose 2 different kinds of seeds. Okay, one seed in which he chose the seeds of a plant which was a which produces true breeding round seeds. which means for many, many generations. When I say true breeding or purebred, I mean that for many, many generations together, all of his ancestors basically have been producing only round seeds. Okay, that was parent one, and parent two, which you can see here, is a plant which is a true breeding wrinkled seeded plant, which means all its ancestors have been producing only wrinkled seeds. Okay, so what he did was he chose both of those as parent 1 and parent 2. He took the seeds of P1 and P2, and yes, he grew them to get the parental plants. Parent 1, which is a round seeded plant, and parent 2, which is a wrinkled seeded plant. Okay, now after getting the parent plants, what he did was, I told you pea plants are bisexual. In order to ensure that self-fertilization doesn't happen, because he wants to try and cross-pollinate between a round-seeded plant and a wrinkled-seeded plant. In order to enable cross-pollination, he had to ensure that they were not eligible for self-fertilization. What he did was, in one of them, he removed the male organs. The male organs or the stamens were removed from this plant. Okay, we call this process emasculation, removal of the male parts. Okay, basically removing the masculine parts of this plant is what we mean by emasculation. Okay, basically just simply cutting off the anthers. That's it, so that it doesn't produce pollen grains, and because of that, this plant has got only female reproductive parts in it. So he went about cross-pollinating by choosing, pollinating between the pollen grains of parent 1, which you can see here, which is the round seeded plant, and parent 2, which we have just emasculated, a cross between the two. And Mendel observed that among the offsprings that this cross produced, all the seeds were, or all the plants produced round shaped seeds. None of the plants produced wrinkled shaped seeds. Okay, this is just an example. Of course, a lot of other steps follow after that. These will be clear to you in the next 5 or 10 minutes. Okay, guys? So for the time being, just understand how artificial cross-pollination is done, and we are good to get started. So here we go. Let's look at it step by step. Mendels experiment steps, or monohybrid experiments steps. Let's now consider the character plant height. Okay, of course, in the example I told you for the artificial cross pollination one, the character we considered was seed shape. Of course, that is also one of the characters that Mendel considered. For this, we can choose any of those characters, but randomly I have selected the character plant height, which has two different traits for it: tallness and dwarfness or shortness. So pea plants can either be tall plants, or they can be short plants. So Mendel raised pure lines with the selected contrasting traits. He selected parent 1, which was a tall plant, a purebred tall plant, which means, I told you the meaning of it, right? Had been producing only tall plants for many generations together. Likewise, he also selected a short plant as the second parent, which had been producing only short plants for many generations together. So he raised pure lines through this method and selected them as the parent generation, which we call as the P generation. Now, P generation is there. The F1 generation, he basically crossed between this tall plant and the short plant. Of course, artificial cross-pollination through emasculation and the method that I have told you. He observed that in the first generation of offspring, or that we call the first filial generation, okay, first filial generation, which we call F1 generation in short, all of them were tall plants. Okay, now children, one important thing to note is that although this image here shows only shows only one pea plant, it doesn't mean only one child was born to these two parents. It doesn't mean that. How many ever daughter plants or offspring plants were produced, be it 10, 30, 40, 50, or even 100, all of them, regardless of the number, all of them were tall plants. Okay, all the F1 plants were tall. So Mendel was shocked. Okay, I chose one parent as a dwarf parent and only one parent was tall. So didn't the short parent did it not play any role at all? How come the children are looking only like one parent? Mendel was very curious, so he went on to carry out step 3, in which he did selfing or self pollination of the F1 generation. So, F1 generation is all tall. He allowed them to self pollinate or self fertilized. This resulted in the second phylyl generation, which we call the F2 generation, and the results shocked Mendel. What did he see? In the second generation, he noticed that among all the F2 plants that were produced, he took the ratio between tall and short plants because he saw many of them were tall, some of them were also short. So he took the ratio between the tall plants and the short plants and saw that it was in the ratio of 3 tall to 1 dwarf. Phenotypically or externally, they were 3 tall to 1 dwarf. Remember, I told you, why am I not writing the genotypes here, children? Because remember, I told you, Mendel, he had no knowledge about the basic structure of the chromosomes or genes or whatever. Not even the term gene had been in use back then. Okay, so all of these results, what we see here is all based on Mendel's physical observations on the garden pea plants. Nothing, he did not use any microscope or anything to come to these conclusions. This is exclusively based on his counting of the offspring generation. So the ratio when he took it, it was 3 tall to 1 dwarf. Wow, how come? So now Mendel was surprised even more because in this particular cross, I told you it was a self-pollination, right? Which means the pollen grains and the ovum are from a single plant. When it's the same plant, it means the parent, the male gametes and the female gametes, both are from the tall plant, the tall parent. Despite the fact that no short parent is involved in this cross, we have got one short offspring. How come? From the grandparent, it's magically inherited by the grandchild, while from this parent, no children could inherit the shortness or the dwarfness. Okay, so what did Mendel do? He just went about proposing his hypothesis. In an organism, he perhaps guessed from his observations, in an organism, there is a pair of factors that controls the appearance of a given characteristic, a pair of factors, okay? An organism inherits these factors from its parents, which means one from each. There is a pair of factors and an organism inherits one from each parent. A factor is transmitted from generation to generation as a discrete, unchanging unit. Okay, this was Mendel's hypothesis. And then he also noticed and guessed perhaps that when gametes are formed, the factors probably separate, because I told you factors are normally found in pairs. Okay, the factors separate and are distributed as units to each gamete. If an organism has two unlike factors, which we today call alleles, for a specific characteristic, one of them may be expressed to the total exclusion of the other. Remember, I told you dominance and recessiveness. Only the dominant allele gets expressed while the recessive allele gets masked in case it's a heterozygous genotype. Okay, so this is exactly why I told you, children, having a thorough understanding of whatever we have discussed in session 2 is absolutely important. All of those terms I will be using very, very often in today's session. But then, for the time being, just understand this is what Mendel guessed based on all his observations on the pea plant. Okay, now, let's just work this out. Mendel's monohybrid cross. Genotypically, let's work this out, because Mendel was perhaps not aware of genes and alleles. As I told you, gene and allele, for both of them, he used the word factors. But now, based on Mendel's hypothesis and based on the knowledge we have today about gametes and alleles, let's try to figure this out using genotypes and phenotypes. Okay, so the parent generation, we said one tall parent and one short parent. So genotypically, because they are purebreds, they are homozygous, capital T, and the short one would be this. The gametes they produce this one would produce gametes of only one type, which is capital T, because during gamete formation, meiosis, as we call it, the homologous chromosomes separate and only one copy of a gene, which means only one allele of a gene enters into one individual gamete. Okay, regardless of the number of gametes produced, all the gametes produced by this parent are of this type, and all the gametes produced by this parent would be of this type. So when these two come together in fertilization, they result in the F1 or the first filial generation, which forms the genotype capital T, small t, which is heterozygous. And I told you, remember, in a heterozygous genotype, only the dominant allele is able to express itself, and this is exactly why all of them were tall. So basically, what we're saying is that although these offsprings had one copy of the dwarfness gene or one allele from the short parent, it couldn't be expressed because the dominant allele was also there in all of them. Now, what happened when Mendel self-fertilized them? Self-fertilization, as I said, the pollen grain and the egg are from the same plant, which means the parents would be, the cross now would be TT and TT. Okay? They would produce two different types of gametes each. This would produce a capital T in one gamete and a small t in the other gamete. This would produce it likewise. And what do you see? Look at the combinations between them. Yes, you have to look at all the possible combinations between the gametes of the two different parents. So this would fetch us a capital T, a capital T, small t, again a capital T, small t, or a small t, capital T, both are the same. 2 into 3 and 3 into 2 are the same, right? Likewise. And here it's going to be a small t, small t. What do you see? Yes, you see that 3 of them have at least one dominant allele, while there is only one which has only the recessive alleles. This is exactly why Mendel found the ratio to be 3 tall to 1 short. Phenotypically, this is what it was. Genotypically, it's a 1 capital T to 2 capital T small t to 1 small t small t. 1:2:1 becomes the F2 genotypic ratio. This is the F2 generation basically. Yes, so here we go, written in a more systematic and a better looking manner for you in the form of a chart. So it's there for your reference. Have a look at it, children. This is the same thing that we have worked out right now. Okay, so yes, this is the conclusion. And you're perhaps wondering what this box is. So in genetics, it's a common practice to, Mendel made it a common practice to use something, a tool which is called the Punnett square, which is named after the person who put forth this, who discovered this square, this type of a systematic way of representing it. Mendel liked logic and liked mixing it with biology, as I told you earlier. So he used this tool to record his observations in the pea plant also. So what you see here is, in the Punnett square, what you do is, along the rows and columns, along one of them, either the row or the column, you represent the gametes of one parent. For example, this is the male parent, the gametes of the male parent represented in the row here, and the gametes of the female parent represented in the column here. Okay, so female and male. And why is it both the same? Because it is self-fertilization. I told you this is the self-fertilization of the F1 offsprings, which we have represented here. Why is this tool important? Maybe for a monohybrid cross, you're thinking that, okay, what we have done here is also not a difficult thing, just drawing arrows and working out the different combinations. But then this is certainly a lot neater, a much neater approach. Moreover, Punnett squares become a very, very important and neat tool when it comes to dihybrid crosses, trihybrid crosses, and so on, because a lot of different types of gametes would be involved in them. Okay, so even for a monohybrid cross, so that we don't go wrong in the cross multiplication or the crossing between two types of gametes of the different parents, it's better to use this. Looks neater also, right, than drawing arrows here and there. Okay, so here we go. This is a so after you write the types of gametes along these two lines, you cross multiply them basically. It's one male gamete and one female gamete crossed together in each of them. This capital T, this is a capital T small t, this is a small t capital T or a capital T small t. As I said, it doesn't make a difference, it's exactly the same, and a small t small t. Okay, so this makes it. Okay, so the phenotypic ratio becomes 3 tall is to 1 dwarf, while genotypically it becomes 1 is to 2 is to 1. How come? It's 1 capital T, 2 capital T small t to 1 small t. But now try to find the relation between these two children, the phenotypic and genotypic ratio. Do you find any relation between them? Yes. The short plant which is there in the phenotypic ratio is the one which is represented in the genotypic ratio also as the last one. But genotypically, what we are writing as 1 is to 2 is together represented as 1 plus 2, 3. Whether it's a capital T or a capital T small t, which means whether it is homozygous or heterozygous with the dominant alleles, it's going to be physically seen as a tall pea plant. That's it. This is the difference between the phenotypic ratio and the genotypic ratio. Okay, and now Mendel went about doing the same kinds of experiments with the 6 remaining pairs of contrasting traits which we have listed earlier. Seed color was one of them, remember, and there was flower color also, flower position also, seed shape, pod color, pod shape, all of that. So if we had to do this for the seed color, now today, because Mendel had done all of this, we know today that yellow seed color is dominant over green seed color. Okay, so let's try and work this out. Yes. So choose the parent, purebred yellow seeded plant and a purebred green seeded plant. The types of gametes they produce would be, this would produce only this type, this would produce only this type. They come together in fertilization to form a capital Y small y, which means all of them, all of the F1 generation would be yellow seeded, all yellow seeded. Yes. Now what does Mendel do? Mendel must have self-pollinated them, which means Y into Y. And what do you see? This is what it is. The self-pollination. Beautifully, you can see exactly the same results, which is phenotypically, it's going to be 3 yellow-seeded plants to 1 green-seeded plant. And genotypically, it's going to be 1 capital Y, 2 capital Y, small y, and 1 small y. Just compare it with the GIF that's here, and it'll be exactly the same. Look at them, happy, cheerful, peace, dancing there. I really like learning biology like this. Okay, so this is exactly the same results. Basically, the values that Mendel has got, phenotypic ratio, genotypic ratio, is all the same as the other example. Yes, so this kept repeating with all the characters that Mendel considered. And based on the monohybrid cross, Mendel came up with 2 laws of inheritance. The first one came to be known as the law of dominance, in which Mendel said characters are controlled by discrete units called factors. What are they? Today we know they are alleles or genes. Okay, factors occur in pairs. Alleles always occur in pairs, right? In a dissimilar pair of factors, one member of the pair dominates the other. What we are trying to say is, in a heterozygous cross, one member of the pair of factors dominates the other one, and hence only the dominant one gets expressed. Yes, this is what Mendel meant by the law of dominance. The next one is the law of segregation, in which Mendel says only one of the 2 gene copies or only one of the 2 factors present in an organism is distributed to each gamete. I have told you that already. At the time of meiosis, look at this. Inside a normal diploid cell, homologous chromosomes are found in pairs. But at the time of meiosis or gamete formation, pairing of homologous chromosomes occurs, but they end up splitting. By the end of meiosis, they split up and enter into different gametes. And hence, this is what you see. Because only one of the 2 allele copies or one of the 2 factor copies enters into each gamete. We also call it law of purity of gametes. Okay? Yes. So, another point I can tell you is the allocation of gene copies is random, which means randomly a gamete can have either the capital T allele here or the small t allele here randomly. It doesn't matter. Okay, there are no hard and fast rules for this. But then overall, even if 100, 200, or 1,000 gametes were produced, say for instance, out of all of them, regardless of the ratio or whatever it is, overall there would be two types. One type having capital T allele and the other type having the small t allele. Likewise for all the other characters that Mendel chose. When an egg and sperm join in fertilization, they form a new organism whose genotype consists of the alleles contained in the gametes. Remember, I have told you this already. When it's a capital T gamete from one parent and a small t gamete from the other parent, at the time of fertilization, they come together to form the genotype of the offspring. This is what we call law of segregation, wherein the gametes or the alleles are segregating or separating out to enter into different gametes. And because the gametes are not mixing up each other with each other when they are being formed, or the alleles are not mixing up with each other when the gametes are being formed, we say a gamete is pure for that particular allele, which is why we also call it law of purity of gametes. Okay, children, law of dominance, law of segregation, or law of purity of gametes. Both of these based on Mendel's monohybrid cross, right? And now, before we go any further, let's quickly check how much we have understood. Okay, 3 or 4 questions, fatafat, right here. Removal of stamen from the flower is termed as dash. Okay, and your time is up. I've told you the answer at the start of today's session. It is emasculation. Yes. Next one. The F2 phenotypic ratio of Mendel's monohybrid cross is F2 phenotypic ratio. And you are right. It is 3:1. Okay. Typically, we represent it as 3:1. But if you write it as, in the rare case, as short, the ratio between short to tall, it would be expressed as 1:3. But normally, the tall ones are expressed first. It's not that the other one is wrong. But then usually this is how we express it. Okay, 3 tall to 1 dwarf. Okay, now, the square made to represent crossing is called dash. The tool that I told you about. Yes, super duper easy. And your time is up. The answer is Punnett square. And well done, children. Yes, children. Now what do we have to do? Mendel's monohybrid cross. We are pakka thorough with Mendel's monohybrid cross. Now we need to understand Mendel's dihybrid cross, which is actually very simple if you understand a few basic details. But before that, I have to tell you a few things. Stay with me, children. Dihybrid cross is coming up after I tell you something that will really, really help you for the remaining months of your academic year. Most of our children have been listing out these problems as the major problems which they've been facing in their current academic year, especially because everything is happening online. Doubts, improperly solved doubts, improper notes, tests and assignments, competitive exam preparation, choice of schedule, choice of language, everything becomes a problem. You're all working from home, we are all working from home, your school teachers are working from home perhaps, and everything becomes a problem. But no, they are not problems anymore. As long as you are associated with Vedantu, all of these are solved. Why is it so? We are also giving you a lot of added bonuses like unlimited live classes, all our micro and crash courses available to you, performance reports so that you can constantly monitor your progress, personalized attention also, because we've got master teachers and class teachers who together work very hard along with every batch to ensure that your progress is continuously monitored. Okay, so all you have to do is just visit vdnt.in/ytpro, and I'll just guide you through the steps here. You just need to choose your grade. Okay, so for you, I'm guessing it's going to be grade 10. And then select your target, which for you guys, it's going to be ICSE 2021. If you belong to a CBSE batch or any other state board, choose it accordingly. then check out all the details. I think I've told you all about the major features, but apart from this, there are also a lot of other amazing features about what is included in what we call Vedantu Pro. Okay. The next step is, of course, click on the button Get Subscription. Now comes the big question. You may wonder how much it perhaps costs. So let me tell you those details also so that you're not confused. We don't want you to leave this session with any doubts in mind at all, because normally a 1 month subscription, which means for 1 full month, if you subscribe to Vedantu Pro, you get unlimited access to all of the features that I have told you for that 1 full month. Okay, the normal amount payable is 2,699, but we are giving you an offer right now by application of the coupon code AMBPRO. Apply this and avail a discount which will reduce your amount payable to 2,294.15. Well, is it even reducing it that much? You may ask, but try doing the math for yourself, children. This is what you are paying for unlimited number of classes, unlimited live interactions, and all the other features that you can see for that 1 full month. Do the basic math for yourself, see how much it would cost you per session, and then decide for yourself. I leave it completely to you. Then comes the 3 month subscription. In case you want to be free of worries for the next 3 months, you may want to go in for the 3 months of subscription, wherein the normal amount payable is 6,999, but the same code AMBPRO reduces it to 5,949.15. Now, children, once again, in 3 months, how many sessions would come? And try dividing this by that. Okay, so you do the math, be smart, do a smart calculation, and see how much it's going to save you. Trust me on this. Just try it out and decide it for yourself. And again, for 6 months, if you want to be basically free of worries for the rest of this academic year, which means till March, I would suggest going for this. Okay, so the amount payable would normally be 11,499, but the same coupon code AMBPRO would reduce it to 9,774.15. Again, for 6 full months, unlimited access to all of these features. I think it's a wonderful thing that Vedantu has come up with. No bias here, but then it's my personal opinion, because we need to ensure that quality education comes to you at an affordable price. So just check it out. This would be the amount payable using the coupon code. If you don't apply the coupon code, you will end up paying at least a little more than this. Okay, 1,000 plus more than what you see here. Okay, so it's completely up to you, children. Do visit the link that I told you. I will share the link once again at the end and decide for yourself. And now let us move on to the Dihybrid Cross, which basically refers to breeding between two plants differing in two pairs of contrasting traits. What do I mean by that? When Mendel wanted to carry out the Dihybrid Cross, what he did was he chose two pea plants as the parents. What was special about them? How did he pick them? Among the 7 characters he chose, he ensured that for 5 characters, they were both having the same traits. Okay, they did not differ in 5 out of those 7 characters. The remaining 2, he ensured that they were completely expressing the contrasting traits of those last 2 pairs of traits. Okay. Now, let us now consider the 2 characters. Of course, Mendel did this for all the 7 characters, but let us now consider seed color and seed shape. Together, he wanted to study the inheritance of 2 genes together at a time. That's it. Okay, seed color and seed shape, let's study them together. So we know that just like the Monohybrid Cross, Mendel ensured that he selected purebreds as parent 1 and parent 2 for their respective characters. Round yellow and wrinkled green. Because they are purebred, we know they are homozygous. Because it's a dihybrid cross, we are considering 2 characters, and that is why there are 4 alphabets in the genotype. Okay, children? Unlike the Monohybrid Cross, where there were only 2 alphabets in the genotype. Right? Here, 4 alphabets, 4 letters constitute the genotype. So, what is it? Homozygous dominant for parent 1, homozygous recessive for parent 2. When they produce gametes, this is how you would find it. Now, this is, children, Listen to me very carefully. In my last 7 years of experience, this is the point where I have seen children getting most confused with. When I was a student, also, this was the point where I used to get the most confused with, because formation of gametes. Well, didn't I tell you that a gamete would consist of only one letter? In the previous one, when I told you monohybrid cross, didn't we say it would be only capital T or small t in a gamete? Why has ma'am written R and Y inside one single gamete? That is because we are studying the inheritance of two genes at a time. Remember, children, two genes in the sense, inside one gamete, we have to consider at least one allele of each of those genes. Okay? For each of those characters, there has to be at least one representation inside the gamete, which means one alphabet representing the seed shape and one representing the seed color. Likewise, here also, one representing the seed shape, one representing the seed color. Okay, it wouldn't be capital R in one gamete, capital Y in one gamete, small r in one gamete, small y in one gamete. No, it doesn't make sense that way, because even if you take the human being's example, 46 chromosomes or 23 pairs of chromosomes in a diploid cell, which reduce in a gamete to 23 chromosomes, 23 unpaired chromosomes. Okay, which means it helps us, it gives us a chance to study the genes which are present in all those 23 chromosomes. But inside a gamete, there is only one allele or one version of each character inside a gamete. That doesn't change even here, which is why, regardless of whether it's capital or small, there is at least one R and at least one Y inside a gamete, right? So this is still going by Mendel's law of segregation or law of purity of gametes, wherein alleles are not mixing up. Alleles of the same gene are not coming inside one single gamete. But for two different genes, yes, of course, they will come together inside one gamete, right? Which is why there is an R and a Y inside one gamete. And your F1 generation, obviously, would be a cross between these two, resulting in a heterozygote. Double heterozygous, as you can see, which means heterozygous for both of these. But regardless of that, you can see that it is phenotypically expressing only the dominant parent's characters. All the F1 plants produce round shaped and yellow seeds, right? Now what? Of course, just like the monohybrid cross, Mendel decided to self-pollinate it, which means now the cross would be between RRYY and RRYY, double heterozygous. Let's try and work this out. Yes, here we go. Okay, so let's start by writing the gametes of one parent along the row, which would be capital Y, capital R, a capital Y, small r, small y, capital R, and a small y, small r. Four different types of gametes which are produced by each parent. Okay, because it's a self-fertilization, the other parent also has exactly the same types of gametes. And small y, small r. Okay, children. So now all you have to do is, when you work this out on your own, just remember this simple rule. Whenever you have to make the gametic combination between the parents of a dihybrid cross, inside one gamete, what should be the components? There have to be two alphabets. There have to be two letters. It can be a combination between a capital R and a capital Y, a small r and a small y, or it can be a combination of a capital R, small y, or a small r and a capital Y. You can see all these four different combinations here. And what would you get? I would actually leave you to do the math, but let's do at least a few of them. Cross multiplication, y, y, r, r, y, y, r, r, y, small y, r, r, y, y, r, r. And this you will have to fill up for all of the remaining boxes. And you will find that this is the result. What do you see? Yes, you would see that phenotypically the ratio is 9 is to 3 is to 3 is to 1. Why so? There are 9 which are producing yellow round seeds. Where there is at least one capital Y and at least one capital R, it means it's producing a yellow round seed. There are 9 of them if you count it. It would be this, it would be this, this, and of course this one, and this one, this one, and this one, and this one. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 of them which produce yellow round seeds. Likewise, do the math for 3, 3, 1 also, which gives you different combinations. Okay, so what is the last one here? It is the completely homozygous recessive phenotype, which is it's producing the green colored seeds which are also wrinkled. There is only one plant among these, which is that. Again here, remember children, very, very important. Although there are 16 boxes which I have filled here, it doesn't mean only 16 offsprings were produced in this one cross. There were perhaps hundreds of babies that resulted in this one particular cross. But then among the 100 different babies, Mendel counted each of them. Mendel noted the phenotype of each of them and took the ratio of them, which is what is 9 is to 3 is to 3 is to 1. Okay, the ratio between yellow round, yellow wrinkled, green round, and green wrinkled. And genotypically, it's a little complex. It would give you 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. The genotypic F2 ratio of the dihybrid cross is normally not asked in exams, but I have put here these values only for your reference. Do it for yourself. Do not memorize this. This is my sincere suggestion. Work it out for yourself and make sure that you understand this concept. Okay, memorizing this is a very, very bad idea. Okay, now coming to Mendel's law, the third law of Mendel based on the dihybrid cross, which is right here. This is called the law of independent assortment, in which Mendel stated that the alleles of 2 or more different genes get sorted into gametes independently of one another. In simple words, what I have written for you in brackets here, the allele a gamete receives for one gene does not influence the allele received for another gene. What I mean to say is, just because gamete, let's number them as gametes 1, 2, 3, and 4 here. Okay, in gamete 1, just because there is a capital Y and the capital R doesn't mean gamete 2 will not receive the capital R or the capital Y, or that gamete 3 and 4 would also not receive the capital Y or capital R. No, nothing like that. It is completely independent of each other. Any gamete can receive any combination of the alleles which are found in the parental genotypes. Okay, and let's read it once again now. The allele a gamete receives for one gene does not influence the allele received for another gene. Yes, so that is it. And also, it another meaning that it has to have is, just because gamete 1 has received the dominant allele for one gene doesn't mean what comes with it should also be a dominant allele. Nothing like that. It can also be capital Y, small r. It can also be small y, capital R. It can be a heterozygous gamete also. Okay, this is also what we can infer from this law that Mendel is trying to say, the law of independent assortment. Okay, children, so very, very important. At a glance, let's go through them. Mendel, based on the monohybrid cross, Mendel came up with the law of dominance and the law of segregation, or the law of purity of gametes. Based on the dihybrid cross, Mendel came up with what we call the law of independent assortment. Yes, children, I hope that really helped you clear up all the concepts you had, all the doubts that you had in your mind about those concepts. And this is a glance at the importance of Mendel's laws. Desired qualities can be introduced, which helps to improve plant quality, of course. Of course, botanists and passionate agriculturists and gardeners would find it very useful. All these basic laws, knowledge of them really helps. It also helps to predict phenotype and genotype of offspring. Now, point 2, I would say it's useful even in humans and other animals. It really, really helps us because these basic rules apply to every sexually reproducing organism on the planet. Okay, and Thirdly, it makes understanding of complex patterns of inheritance very, very simple. Okay, children? So there we go. Let's quickly answer 3 or 4 questions very, very quickly, and then we will be ready to wind up today's wonderful session. Now, dash is the F2 phenotypic ratio of a dihybrid cross. F2 phenotypic ratio. Okay? And your time is up. Yes, it is 9 is to 3 is to 3 is to 1. The order in which we write it is like this: 9 is to 3 is to 3 is to 1. Yes, next one. Which of these Mendelian laws is also known as the law of purity of gametes? Yes, I have told you the meaning of law of purity of gametes. Is it the law of dominance, law of segregation, law of independent assortment, or is it both B and C? Your time is up, and the answer is law of segregation, wherein alleles of a gene segregate and enter into different gametes, meaning that gametes are pure for an allele. Okay. The F2 genotypic ratio of a monohybrid cross is dash. F2 genotypic ratio. Okay. Monohybrid cross. And your time is up. The answer is 1 is to 2 is to 1. Yes. And the next one. In the genotypic ratio 1 is to 2 is to 1 of the monohybrid cross, how many are true recessive, or how many of them are truly recessive? Okay, and your time is up. The answer is 1. In 1 is to 2 is to 1, true recessive refers to whatever was genotypically and phenotypically recessive. There is only 1 in this ratio which is like that. Yes, and well done, children. Whoever got all the answers correct, great job and great going. This actually means you have managed to understand the 95% of the hard concepts of this chapter. Okay, if you have paid attention to me in the last 40-45 minutes, yes, things are very easy for you now, and you now know what is monohybrid cross, what is dihybrid cross, the 3 laws of Mendel. Do not leave. I have to give you the homework question also. But before that, let me remind you, remember to visit vdnt.in/YTPRO and use the coupon code AMBPRO. And yes, this is your homework question for today. A pure tall plant bearing red colored flower was crossed with a pure dwarf plant with white flowers. Given that tallness and red flowers are the dominant traits, what would be the nature of offspring in the F1 generation? So, physically or phenotypically, what would F1 offspring look like is what you have to tell me. Okay, so work this out. Work out all the possible combinations of the different traits that Mendel did. If you are interested, well, I have done that as a student because I started loving this subject the moment I understood the basics very clearly. Okay, so you can also go ahead and do that because it will just strengthen your basics in this concept. Okay, children, so thank you very much. If you have enjoyed this session, please do remember to hit the like button right now, because otherwise there's no means of me knowing that this was useful for you and that you have enjoyed this. Please do share it with all your class 10 ICSE friends, all your class 10 CBSE friends, and all your class 11 and 12 CBSE NEET aspirant friends as well, because this will benefit all of you guys that I have mentioned. And in case you've not subscribed to our channel yet, Vedantu 9th and 10th English, please do hit the subscribe button right now, and of course the bell icon right next to it, so that you do not miss out on any of the notifications that you might possibly get from us, because every single day we keep coming up with newer and better content just for you. Yes, children, until we meet again, this is Ambika signing off. Take care, stay safe, and bye-bye.